Next is Duncan Irshcick from UMass Amherst. And again, as a reminder, you can add a question at any time in the Q&A, and all of the speakers here in this session will be available to answer your questions afterwards. So Duncan, can you share your screen?
Yep. Okay, can everyone see? I assume everyone can see that, hear me okay?
Yep, look good, sound good. Thank you.
Thank you. Great. Thanks so much for having me. This has been exciting. I want to talk today about some work on novel 3D tools for reconstructing living animals. That's been a focus of my work for the last several years. As this whole meeting is going to really outline, 3D scanning as a form of, if you want to think about it, digital heritage, is a fascinating enterprise and has many facets. There's obviously aspects of digital 3D photogrammetry, which we'll be talking a lot about today, but there's also other areas, right? There's laser scanning and white light scanning. There's CT and MRI scanning. For those of you, obviously, who've ever had the unfortunate circumstance of being told they have to have a CT or MRI scan, you're familiar with the concept that you go to a hospital, they take a scan, they create a 3D model. And so there's a long tradition of creating scans of dead objects, such as the skull shown on the left, or a brain, or a historical artifact such as shown on the right. But there's a lot less tradition given to the concept of reconstructing what a living animal might look like so that we can study it for the sake of science. Let's see here. Why is it? There we go. And, I don't really have to sell this to your group, but I think it's important to remember some of the advantages of 3D scans when we think about a living animal, which is they provide a 3D map, shape map of an object or organism. So they really do provide that ability to understand the shape of an animal in a new way. And this is going to become incredibly important when we think about certain species that really aren't found in very well good systems in natural history collections. I'm not going to be talking about today, but I'm going to be talking later on in other talks about how we have some new work also that using 3D photogrammetry scans is providing a 3D map of the color of an organism. And we can map that onto the shape. These can be modified to add movement and to study locomotion. Another focus in my lab I won't be talking about today, but is exciting for the future. And the final result can be strikingly realistic. And this is very important when we think about megafauna or animals that we examine in terms of demographics, because demographics were interested in studying particular animals and following them over time. So what about living animals? As an example of this is Mac. This is a female leatherback sea turtle that we studied in Juneau, Florida. We reconstructed this animal using 3D photogrammetry, so the body shape and color is accurate. What I admit is a bit of a flavor on our part was simply the movement that we added to Mac. But the shape was correct, and that's because we had the fortune of being able to work alongside the researchers who study this animal and have access to this animal as well. And so what could you do if you had these kind of shapes of 3D animals? And imagine as scientists, we had access to these for different kinds of species. Here's a list of some of the things that our lab has thought of, but I'm sure you have your own ideas. I'd love to hear them. You could certainly collect 3D models of organisms in the field. You could get their DNA. You could release them. This is becoming more and more commonplace in regions of the world that we work in Africa and Southeast Asia, where a lot of countries don't really want you euthanizing or killing or depositing specimens or exporting them. You could perform computational fluid dynamics on 3D models of flying and swimming animals. There's a long tradition of this, but this typically has been done on kind of rough caricatures of species and not accurate models. You could use the 3D models to estimate body condition, animal health. This is one of the biggest areas of growth for a lot of our models. You could design new kinds of robots, and you could use them obviously in educational portfolios. So the 3D photogrammetry is something you'll be very familiar with. This it's already been talked about. But the idea of taking a series of photos of an object, in this case, this is actually a golden poison frog at the Zoo Atlanta with tadpoles on his back. You take a series of 2D images, you convert to a 3D mesh, and then create a 3D texture geometry. And we've been focused on creating rigs and systems that can work with living animals in the field or in the lab Our initial focus was on small reptiles and amphibians, but we've expanded to many other animal species. You can see some of these concepts down below with some of the students I've worked with, like Dylan Briggs, Casey Smart, and Zach Corbeau. But I'm going to be talking about two larger animals that are really not that we've been interested in. One is megafauna, specifically in Africa, because we've been interested in recreating these animals to understand health and body condition. This is a proof of concept that we did to recreate a 3D southern white rhino to develop novel body condition metrics. This is Bakari. Bakari is a southern white rhino in the Perth Zoo, a very mellow rhino. We were able to actually hook up a series of cameras around this cage while it fed, and then get a really good 3D photogrammetry profile of the animal. From the 3D mesh, we could create a final 3D render with color that was an accurate metric of its shape. With the vet staff at the Perth Zoo, we were able to then measure a whole series of metrics, or they were able to do it, I should say. This is a very well-behaved rhino. I don't recommend any of you ever try this with a rhino in the wild. Body height and body length. And so we had a series of morphometric measurements that we could use to scale the model. We knew the mass of the animal at the time. And this allowed us to ultimately create an accurate 3D model of this rhino. Again, the motion is something that we added, but the body shape, the texture, everything is accurate. And by the way, I should say that we have a lot of knowledge and skill about recreating models, and we work with lots of different partners around the world on this. So if you have a project you're interested in, please come talk to us. So the concept here is to estimate body condition from volume mass relationships. So we hope to scan other rhinos or to use this 3D model to create other digital copies. From this, we know the mass and the length of the animal. We can calculate the volume and the surface area, which is one of the exciting parts of this work. And from that, we can ultimately create new metrics for body condition that we hope to then use machine learning systems on in the wild to estimate how much these animals weigh. The second part is underwater. We wanted to develop methods to do the same thing, but with underwater animals, and we focused on hammerhead sharks, which live in Bimini and will dive with. You can dive with them fairly safely. There's a person in the middle who feeds the sharks. We have a series of cameras around. You dive with the shark, you get a series of photos, much like the rhino, and create a 3D photogrammetry scan. We weren't so interested in the nurse sharks around there, but you can see the big hammerhead shark in the middle. From this, we were able to focus on one particular individual. Her name was Nemesis. She's about 10 feet long. She's a female shark, and we're able to reconstruct the accurate body mesh. And these sharks, by the way, grow very large. They grow up to over 20 feet long, although in the wild, we rarely see animals that large. So this kind of body mesh, these animals aren't easily found in natural history collections. And this is, again, the 3D model of the shark. We added in the mouth motion and other motion, but the body shape is accurate. From this, we're actually able to actually ask some really interesting questions. One of them is that when you look at different animals we've scanned sea turtles, we've scanned marine mammals and sharks, and many other species. This is a plot just for aquatic species comparing volume and surface area. And what you see is the isometric slope should be about 1.5, and the actual slope is 1.48. So despite the fact that we're comparing reptiles, marine mammals, and fish, elasmobranchs, these animals all show the same basic relationship between volume and surface area, which is really fascinating. And we're going to continue to add to this plot. We put these models online. They are open access. At last count, I believe, actually, I didn't have time to update the slide. I think we actually have close to 180,000 downloads. They get used for all kinds of applications, from education to scientific. If any of you want to create your own model of a living animal, please come contact us. We are a cost center, so we can actually work with partners all around the world to create new content. But we do allow people to download them for free for nonprofit use. And it's been really exciting to work with lots of different people on that front. And I'll stop there. Thank you so much.